Okay. So thank you very much. I'm presenting on the title, the 16SR Group 22 Phytoplasma in palms, insect, and alternative hosts. And this work has been conducted by my colleagues in Ghana and our collaborators in the UK at the University of Nottingham. Sorry, it's not moving. Just a moment, my screen is not moving.
We don't know how to help.
My slides are not moving, but I'll get it sorted out.
Maybe you can upload again, I don't know.
Okay.
Ndede, you could stop sharing and then try share again.
This is okay.
Okay.
So do you have it open on your screen? Do you have the presentation open on your screen?
Yeah, it's open on my screen.
All right. Okay, we are seeing it now. Go to the beginning.
Okay.
Go up and press it first, the icon on the top in the red band. Press the icon, the first icon on the top on the red band.
Yeah.
Or go down to the bottom, Ndede. Yes, then where you are, that's okay.
Okay.
Oh, yeah, it's okay.
Thank you. It's okay now. It's okay now. Thank you very much.
You're welcome.
So this will be the outline of my presentation. I'll talk about coconut cultivation in Ghana, the symptoms of the later yellowing disease, the diversity of the group 22B phytoplasma in Ghana, and then I will end it with the hosts of the phytoplasma, that is alternative plant hosts and insects. So by way of introduction, coconut is a very important cash crop along the coastal belt of Ghana. And this is particularly so because in the coastal areas of Ghana, the soils are poor and not many crops can grow there, but the coconut thrives in this area. The palm supports about 4.2% of the national population. And the products are used by all sectors of the society. If you look at the coconut oil here, the top one is a product that can be purchased by the affluent in society. This one can be purchased by the middle class, the working class, and this is the village level technology. So the products are can be all sectors of society use the coconut products. The coconut palm and the coconut industry is, however, threatened by a later yellowing disease. The disease was first observed in 1932 in the eastern part of the country, and so far it has destroyed about 20,000 hectares of plantations. Now, these are the symptoms of the disease. In berry palms, when the disease starts, there is a premature nut drop. There is progressive yellowing of the front, starting from the oldest front and progressing to the younger ones. And then on the terminal stages of the disease, the crown falls off and we have the characteristic telegraph pole stage of the disease. Now, the pathogens, the later yellowing disease in Ghana is associated with the group 22 subgroup B, phytoplasma. And in the group 22, there are two main subgroups, as Assunta mentioned in her presentation. We have subgroup A, which is encompasses the pathogen that causes is associated with the disease in Mozambique and Nigeria. And then subgroup B is associated with the disease in Ivory Coast and in Ghana. Now, to study the diversity of the phytoplasma in Ghana, under this Tropicsafe project, we have conducted surveys all around the country to detect disease presence. And we have done so using extensive and intensive methods. We have used vehicles to assess all motorable roads, and farms where the vehicle cannot pass, we go on foot to inspect the individual infected palms. And then we have also collected over 100 samples across the country in all the major coconut growing regions of the country. Now, what we have done was first to use nested PCR to confirm the symptom observation we have seen in the field. And we have used nested PCR using 16SrXXII and then US-based assays. And from the result, we realized that largely the symptoms observation, the symptoms we observed were consistent with the PCR results, except in the early stages of the disease, that is at the symptomless stage, where some of the, where although they were symptomless, nested PCR was able to detect the phytoplasma in the samples. Apart from that, the symptom observation and the PCR analysis were congruent. Now, we have sequenced for all the positive tests, we have sequenced the samples. And based on the 16S ribosomal RNA-based gene, we didn't find any sequence variation. All the sequences were uniform. However, with the US-based assay, we found two SNPs that differentiated the samples we collected from the Western and Central regions from the samples we collected from the Volta region. So this is the map of Ghana, and these are the three main coconut growing areas of Ghana. We have the Western region, the Central region, and the Volta region. So we have a strain that is specific to this area of the country, and the other strain is specific to the Volta region of the Volta region. Now, we have also investigated the alternative other plants apart from the coconuts that can host the pathogen. Now, in Ghana, there are several areas where the epidemiology of the disease seems to suggest strongly that there are alternate hosts are involved in the spread of the disease. One particular place is the research field at a location called Cape 3 Points. At this place, is a research field. We have planted successively. When we plant coconut, the disease devastates it. So it is hypothesized that in the intervening period where there is no coconut, the phytoplasma will be harbored by some other host, either an insect or a plant species. However, so in Ghana, typically, this is the kind of coconut fields you have. You have coconuts, but we have other plants that are grown in the coconut fields, which are very suitable, and so a lot of vegetation which can easily host the phytoplasma. However, previous studies to identify alternate hosts have failed. We have not been successful in identifying alternate hosts of the phytoplasma. Under this project, we have done extensive sampling, and we have sampled 24 plant species from 18 plant families. And then the samples were collected from 12 locations in the three main coconut growing regions of Ghana. And there's a map of the place where we have sampled along the coast from the Western region right up to the border with Togo. And the sampling has been carried out from August 2017 to June 2018, spanning both the seasons we have in Ghana, that is the rainy and the dry season. And these samples have been analyzed using nested PCR and sequencing. And what we have done is also to have shared the samples with our collaborators in the UK. So we have done independent analysis of the samples. So this is some of the plant species that have been sampled and tested for the ability to host the alternative, the phytoplasma associated with the coconut disease in Ghana, different array of plants. So for results, we were able to identify or we found the phytoplasma in 3 plant species. That is Laportia estuans, Stachyphyta indica, and Petodon petandrus. And these are the various, the number we have detected as positive in the total number of samples we have tested. Now, all the plant species that gave were positive for the phytoplasma were sampled from the central region. None of the plants sampled from the western and Volta region harbored the phytoplasma. Now, we did sequencing of the samples, and we observed that the phytoplasma strain that was found in the alternative host plants were the same type associated with the disease in the central and the western region, as not the Volta region strain. So these are the pictures of the alternative host plants we have identified: Stachyphyta indica, Pteroda petandrus, and Lapotia estuans. Now for insect hosts, now the vector of LYD in Ghana, like in many other places, is not known. In this project, we have done insect sampling from devastated, disease and disease-free fields. And we have also carried this sampling in 20 locations. Sampling has been done between August 2017 to June 2018. And so this is, for example, this is a devastated field we have collected insect samples from, and this is a disease-free field we have collected insects from. Now, from the locations, we have categorized the insects into the predominant species and the species and the other species. So in all, about 2,000 insects were collected, and we have analyzed them using nested PCR and sequencing. Now, the results, we found positive tests for 2 species of Distrombus and 1 Hepatara amata and Hepatara amata. Now, for this project, we could not do transmission trials. However, we studied the population dynamics of the putative vectors. And so we have used circuit traps to monitor the insect populations. And we have conducted this experiment at 2 locations, one in the central, Agona Panta in the western region, and the other in the central region of Ghana. We have also included the putative vector of the later yellowing disease in Côte d'Ivoire, which is the N. Keta, because it is abundant in our environment. However, in our in this study, we sampled a lot of the Ndede Keta, but they were not positive for the phytoplasma. And we generally observed that in the western region, the N. Keta among the suspected vectors was the most abundant. The ones we found in this study where the numbers were very low, we monitored their numbers in various palm types. In the SGD palm, these are just a sample of the results in the West Africa tall and in other palm types. So the numbers of the suspected vectors that we have found positive for the phytoplasma, the numbers were very low. And when it comes to the central region, generally all the insect population for both what we have found and the one that occurs in the Ivory Coast were very, very minimal. The highest we could find was about 2.5 insects per trap per month. So generally the insect populations of these suspected vectors are very low in our environment. Okay, so in summary, we have identified 2 strains of the group 22 phytoplasma in Ghana. One that is specific for that one associated with the disease in the Volta region and the other in the central and western region. And this this finding is very important, and it will influence how we conduct future resistant trials and in the management of the disease. We're also able to detect the phytoplasma in alternative host plants. In previous studies, we have used single-run PCR, but in this study, it was nested PCR that was able to use to detect the phytoplasma. It therefore suggests that the titers of the phytoplasma in alternative host plants will be very, very minimal, because in coconuts, the phytoplasma is readily amplified using single-run PCR. And for the alternative host plant that we have identified, we'll incorporate them into our disease management strategy. And the knowledge we have generated from the population dynamic studies will influence how we conduct our transmission trials in future. So thank you very much for your attention, and I look forward to answering your questions.